episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome everybody again to another episode of our show with another really fascinating guest for you today, helping to create a better tomorrow on many different fronts. Today we have the honor of being joined by Howard Leonhardt, who is the founder of Leonhardt Ventures, which is the world's first innovation accelerator that is focused on the convergence of bioelectric and biologic technologies for a range of opportunities, both organ regeneration and healing. Howard is a long-time accomplished inventor and serial entrepreneur, dozens of U.S. patents, over 100 patent claims for his various products in treating cardiovascular disease. He has dozens of new patent claims pending. His talent, or Taheri-Leonhardt stent graft, which was developed in the early 1990s, still holds a leading world market share for repairing aortic aneurysms without surgery. And his inventions to date have been involved in treating over 500,000 patients around the world, 60 different countries. When he's not focused on all that, Howard is also the co-leader of Startup California and founder and chairman of the California Stock Exchange, which is preparing to be the first social good impact stock exchange, currently operating the Cal-X 30 Social Good Impact Fund. He founded Cal-X Crowdfund Connect, crowdfunding campaign management company, and Cal-X Stars Business Accelerator, a business incubator and accelerator focused on cardiovascular life sciences and social good impact innovations. In his current portfolio, he has over 30 medtech and regenerative medicine startups. His network's put together over $150 million to date, including technologies including first implantable, programmable, and refillable stem cell pump, multiple different tools for brain, eye, and aortic regeneration, and another of other organ regeneration spin-offs. Howard also serves as the state spokesperson in California for the Jobs Act and crowdfunding for Startup California. Has given dozens of speeches on this subject, and he's operated Leonhardt Launchpads, North Cal, at the University of North California Science and Technology Innovation Center since 2008. Recently opened Leonhardt's Launchpad Utah in Salt Lake City, just near the University of Utah, and he has served on the board of directors of the University of Northern California since 1999. Howard Leonhardt, thanks so much for taking the time to come on the show today.
Well, you did a good job with the complex introduction. Thank you very much.
Most definitely, Howard. We were chatting beforehand. You and I have known each other for a couple of decades now, but I'd love to start things off like we typically do. Obviously, you've had a fascinating career to date. If you could take us on a short journey, sort of from the beginning, sort of go a little bit into sort of where you grew up, how you got interested initially in sort of the area of medical devices and sort of the beginning of this path. And we'll get into regenerative medicine and bioelectronics a little later on, but talk a little bit about the beginning, if you would.
Thanks, Ira. And thanks everybody for listening today. For myself, I graduated from a small tech college in Minneapolis called the NOCO Technical College with a diploma in international trade, unusual pathway into medical device engineering. I started off helping manufacturers of cardiovascular devices to build their export markets. And my routine would be I would drop off the samples of the newly developed products by these small companies, and then I would circle back. This would be Saudi Arabia, Indonesia, South Korea, Taiwan, places like that. And I would come back a month or 2 or 3 months later and make my route of all the investigational centers that I was working with, and I would get their feedback. And after a while, I would bridge between the doctors and the engineers. I'd get back to the small company factory and I'd say, if we want to sell more of these, you got to fix this and fix that and got to make this better. And the doctors would really like to see this built into the next design. And after a while of bridging that gap between the physicians and the engineers, I started to kind of have my own ideas about how to improve the product. And that started off with our very first product, the Polycath cardiovascular balloon catheter. There's a little bit more to the story. Before we started manufacturing and developing, we started to private label, where we had one of those small manufacturers make the product under our name. So it said manufactured for World Medical Corporation. And everybody thought we were the manufacturer because the print of manufactured for instead of manufactured by was very small. And then I started to feel like a manufacturer because all the physicians treated us as if we were making the product. And in a sense we were, because we were hiring somebody to make it for us. But our supplier was bought out by B. Braun of Germany, and it was called Nova Medical Specialties in New Jersey. And the turning moment for my entire career was the moment when I called B. Braun, who now bought this small company. And we were one of the biggest customers for Nova Medical Specialties, buying more than 20,000 cardiovascular balloon catheters a year, and they only sold about 100,000. So we were about 20% of their business. And the new buyers were giving us a lot of trouble. And they basically were saying, well, we're going to stop your private label contract. You got to now take the B. Braun package. And we were, no, we don't want to do that. And we told them we were right in the middle of working with the engineers of Nova Medical Specialties to change the balloon from latex to polyurethane because many patients were allergic to latex. They said they didn't want to do that. And we had some other changes that the doctors really had been asking for. And they again said, we don't want to do that. And it was a frustrating call. And basically I hung up with a little bit of temper and said, how hard can it be to make them? Let's make the damn catheters ourselves. So I got a teacher, and he taught me how to make balloon catheters, and I entered manufacturing of cardiovascular balloon catheters. We did all the tests to get through the FDA, and that was the beginning of that journey. And the part of the story that maybe gets more interesting is along the way, we decided, well, we're not making a lot of profit on these cardiovascular balloon catheters. We got to find a way to leverage our core competency to have a higher margin product. And that's when we developed the Talent Stent Graft, the Taheri-Leonhardt stent graft for repairing aortic aneurysms. At the same time, we developed the first percutaneous heart valve. And we took both of them out to surgeons to say, hey, we want to repair aneurysms and we want to repair heart valves without surgery with this catheter that's snaked up from your groin and releases the valve or releases the stent graft, the same ones that you guys are sewing in place after you crack open the chest and break open the ribs and clamp the arteries to be able to hand sew these products. We're going to hold them in place with a strong nitinol spring. Well, every doctor we showed it to said it was a terrible idea. They even became angry with us because we're going to take away their job of doing surgeries for this. And we had a large investor at the time, Nippon Zien Company of Japan. And after I presented to the top Japanese surgeon these ideas, and he absolutely ripped them apart and said, you know, they're never going to fly. You're going to cause defacing of the company because you're going to have disaster in the marketplace. And Dr. Marty Matsuo puts his arm around me on the way out, and he said, you know, we told you to focus on cardiovascular balloon catheters. We invested because we were making a really good cardiovascular balloon catheter. You have a defect rate of 0.002%, and we want to get it down to 0.001%. So focus on that. And we actually set aside those two projects because we thought, maybe we had a good idea, but it looks like it was a terrible idea. Race forward, last year, it was announced that more heart valves were placed with catheter-based procedures than surgery. Race forward, and more aneurysms are repaired with catheter-based procedures led by our device. But we lost 3 years. So we threw all those ideas away in 1991. And then in 1994, we saw some others coming with the same ideas. And we had to jump back in 3 years later and catch up now to two other companies that actually were ahead of us. So there was a lesson to be learned there, is the so-called experts are not necessarily going to embrace something that is going to change the way they do things. We now try to, when we take a new product out, if we get negative response, we're kind of thinking, oh, we probably got something here.
Excellent. And, you know, obviously, you know, you had a lot of this success with the catheter, learning the cardiovascular space. You moved then, you know, obviously about 20 years ago, stem cells started getting hot as an area. You get involved in that area, and then you learn as you're going along in the stem cell space. It's had its ups and downs over the last, say, 20 years, but that stem cells are extremely important and they do a lot of exciting things, but you began to learn that there were other important components of sort of regeneration, which is a very complex biologic process. Enter the domain of bioelectronics or bioelectricity or electrophysiology. There are a lot of different terms here, different concepts that go back about 100 years, but which are sort of starting to peak up now in the 21st century. A couple of months ago, we had Mike Levin on from up at Tufts, who's very excited about the space. He works more in the area of how you can develop drugs to modulate ion currents and so forth. Talk a little bit about your history in the stem cell space, and then what got you interested in looking at some of these adjuvant technologies like bioelectricity as a component of sort of the big picture of regeneration.
I'm going to have to look up your episode with Michael Levin. I know Michael, we've hired him as a consultant in the past, and
Great guy.
We greatly admire the work. In fact, we have to admit, we learn a lot from the work that he has pioneered. He got the grant from Paul Allen, yep. And he really teaches all of us about mechanisms of action. And when I went to visit us, I asked them, is it our bioelectric signaling that's communicating with the DNA to build a protein, or is it communicating with the cell membrane to open and close pores? And he flippantly answered yes. And it was a either or question. Yep. And then he went on to explain that it depends on the situation. It's a little bit of both. And I greatly admire his work. And I'm going to look up your podcast with him. I didn't see it yet. For us, it's this dual path of biologics, which starts with stem cells, as you mentioned, and bioelectrics. Mm-hmm. Collaborating with Dr. Ray Scow and Dr. George McGovern in Pittsburgh, they did it, but we joined with them after they did it. They injected muscle stem cells for the first time to repair an infarcted heart in large animals. They did the work in 1988. It was published in the Physiologist in 1989. Shortly after the publication came out, we met each other, and I signed both of them up to our team. So from that point forward, we have been working together. In fact, George McGovern Sr. has passed away, but George McGovern Jr., his son, is now head of cardiac surgery at Allegheny Medical Center, and he is consulting with us. But that was 1988 and 1989, and for me, the whole journey began. I was flying to Brazil to do some heart valve cases with LabCorp Laboratories in Belo Horizonte, Brazil. That was just prior to the percutaneous heart valve. We were actually placing them surgically in those days. And two things happened on that trip. One is that on the way there, I was reading Scientific American, and they talked about fetal endothelial cells having a 1,000 times higher proliferative rate than adult stem cells. And that the future of medicine perhaps was going to be regenerating arteries. And that kind of lit me up. I just thought that, the body's made of cells, and the way that your heart valves, your arteries, your heart, any organ is originally made with the convergence of bioelectrics and biologics, stem cells, yep. proteins. And it just made sense to me to try to rebuild them more close to the way that they're built in the first place than to do what we had been doing. I was on my way flying to Brazil, and we were going to implant a piece of cow tissue that we cut to form into a heart valve. And that's very artificial. That's not part of your body. And interesting enough, on that trip, I went to go see the heart valve put in by Dr. Domingos Moraes, the first surgeon in Brazil who had ever done a bypass surgery. Mm-hmm. Even in 1988, he was already getting up there in age, because he did his first bypass surgery, I think it's 1965, around the time when the U.S. got started on that as well, 66, 67, sometime around then. But the interesting part of the story was I go to Belo Horizonte, they give me the valve, and they say, go to Rio de Janeiro and bring this to Dr. Moraes. He's going to do a surgery, and he'll give you a chance to see a surgery, because I actually hadn't seen one before, even though I was selling these heart valves and I was the export representative of LabCorp Laboratories. Mm-hmm. And so I bring the valve, I give it to him, he puts it on the shelf, he cuts open the patient, breaks open the ribs. The first thing I thought was unusual is they go over to an ice bin like you have in the hotels, and they scoop up a bunch of crushed ice and they pour it into the chest. And I thought, wow, that doesn't seem that sophisticated. But he explained that they're trying to slow down the flow. And then he goes in there and he's moving his hands around, and all of a sudden he starts to close up the chest, and the heart valve is still sitting on the shelf. And I'm went there to go see the heart valve put in, and I'm thinking, oh my gosh, this surgeon is so old that he forgot to put the heart valve in. And so I was going to tap him on the shoulder and say, oh, excuse me, you forgot something. It's like, it's still there on the shelf. You didn't put it in. But I didn't do that, of course, out of respect. So I waited, and then we go wash our hands. And I said, hey, Dr. Marias, you didn't put the heart valve in. And he says back to me, how are you going to learn something today? And I said, what's that? And he said, heart valves, like all organs, are a lot like your wife. You're a lot better off keeping the one you have if you can. And he went on to explain that a lot of times he would get in and he would see just a little bit of calcification, and he would scrape it off with a needle scalpel, or not a needle, a scalpel blade. And if he could clean it up and he saw that the heart valve closed, he would rather preserve the person's own heart valve instead of putting in a pig or cow or steel. All of those have problems, especially in those days. Or plastic valves that were coming out at the times. And he said, all of those artificial things are never going to work as well as your natural body organ that you were built with. And if you can preserve that, that is a lot better way to go than to put in any of these things that we're doing. Despite the fact that LabCorp wants to sell some heart valves, he's not going to do it to get their sales up. He's going to do what's best for the patient. Well, I learned something from him, and he kind of set the course of my work for the rest of my life, that in very simple terms, the rest of my life after that is trying to be dedicated to help people keep their own organs and regenerate them instead of getting an implant, despite the fact that, most of our wealth in the past has come from developing and making implants. I really get away from that.
And, obviously when you look into your portfolio currently, many exciting projects in cardiovascular, brain. You have some cancer work going on, then a lot of, as we talked before about sort of the cosmetic side of things. This is just how does it typically work? 'Cause I know you'll work with a lot of universities also. You have research, UCLA, University of Utah, University of Minnesota, many other groups, Texas Heart Institute, the cream of the crop here. How does your incubator system work if, I'm researcher X that's coming along and have a good idea? What happens in the process? How do I become part of Leonhardt All Stars or the the team and get involved in the projects?
Thanks for asking that. In a simple term, Leonhardt's Launchpads, which has merged with Cal-X Stars Business Accelerator. Leonhardt's Launchpads founded in 2008, as you mentioned. Cal-X Stars Business Accelerator in 2013, and in 2015, I think we merged them, and we just call it Leonhardt's Launchpads now. And the concept was to focus on a convergence of bioelectrics and biologics. And really, in very simple terms, it's one stimulator that we have made for us by Mettler Electronics in Anaheim. Okay. It has a 510K market clearance for improving circulation and basically muscle healing, as well as pain relief. So if any application that we apply it to falls within those categories, it actually is FDA cleared to be sold. But we try to broaden the indications of use. So we have a core IP. It started with stem cell homing. So if we bioelectrically stimulate your knee, we can get your stem cells to migrate from your bone marrow to your knee. If we stimulate your heart, we can get them to migrate from your bone marrow to the heart. If we stimulate your brain after a stroke, we can get your own stem cells to migrate to your brain after a stroke. So that was kind of our crown jewel. First major patent is bioelectric stimulation expression of STF1 and PDGF, which are known to be stem cell homing factors. Okay. Then we added a whole suite of muscle regeneration protein expressions: follistatin, klotho, IGF-1, limb muscle. Then we added a full suite of angiogenic or arteriogenesis. In 1999, we published a paper in Circulation on bioelectric stimulation for growing new blood vessels via the controlled expression of VEGF. We were so excited about the paper. We had really good results. Later, we learned that there were flaws in that design, because VEGF created vessels were leaky and they didn't last over time. They would kind of retreat and they would leak. So later we learned that you had to get the right sequence. You had to do VEGF followed by STF1, PDGF, perhaps HIF1 alpha, nitric oxide synthase to dilate the vessel. And if you did all these things in the right order, you would get true arteriogenesis, blood vessels that had a true endothelium lining that lasted over time. So we call that our arteriogenesis suite. And I left a couple of the proteins out of that description. And then we later developed, which was bigger challenge, the nerve regeneration suite, which is highlighted with sonic hedgehog, again, IGF-1, limb muscle again. And we have a couple of others that are in the works that we haven't perfected yet, so I'm not going to mention, but we're about to, we think, add to our nerve regeneration. So then we had spent 20 years and over $100 million learning how to regenerate the heart. In 2008, Bioelectric went public, $76 million IPO on NASDAQ. I had resigned as CEO about March of 2007. We went public February of 2008. The only U.S. biotech IPO in the whole year of 2008, by the way. And we had brought in Bill Pinion from J&J. He just looked better for Wall Street. I'm kind of rough around the edges, and I'm a technology guy, and I don't really enjoy the whole Wall Street thing. We thought he would be a better spokesperson for the company with that financial community, and he was. And but by that time, basically, as it often happens, the VCs, the money funds had basically taken voting control of the company. And typical of form, me being the technology guy and they being the business people, resonate with the Steve Jobs story, even though I don't necessarily compare myself to him. We had a clash about the future. I wanted to bring bioelectric stimulation. I wanted a pump that we would refill with stem cells. And not only stem cells alone, but I wanted to mix the stem cells with an egg yolk-like mixture, gelatin with a bunch of nutrients. What they were doing was running a catheter up and injecting just muscle stem cells in scar tissue of the heart one time. We had done studies showing if you came back and injected a second time and third time, which we published in the European Heart Journal, you would get more muscle formed in the scar tissue. We also published studies at Cleveland Clinic and University of Florida that if you added certain proteins like STF1 or Connexin 43 or eNOS, in particular, if you put them into a hydrogel, you would get better results than just naked stem cells alone, because a lot of the stem cells died. And then we discovered that it would be better to have a pump under the skin that you could refill once a month or once a day than it would be to go to the cath lab every month for 36 months in a row to make a new delivery of stem cells. So basically, there was a big argument at the board level. I wanted to go that way. In fact, I was arguing that before the IPO, and they basically put a muffle on me, said, on the IPO roadshow, don't mention a thing about the bioelectric stimulation work, don't mention a thing about hydrogel, don't mention a thing about the pump, and just focus on what we have now, what's in clinical trials now, one-time injection of stem cells, and say, this is the greatest, let's go forward. And it was very good, and they had very good results. So, not to disparage those guys, in that clinical study that they were behind, double-blinded, randomized, placebo-controlled, the treated patients improved 91.7 meters in 6-minute walk, and the placebo patients declined -4 meters. 84% of the treated patients improved. Only about 16% of the procedural patients improved in whatever category you looked at, heart failure class or exercise capacity. So, they had reason to get behind their plan. It was pretty good. But I just was focused as a technology guy and making it better. And again, shorten up the story, which is hard to do, but we basically, that next year after the IPO, we had a divorce of sorts. And they owed me, in my calculation, $4.7 million. I had borrowed the company money that I had made from my stent graft success before. They didn't pay me wages. They didn't. We had a scientific advisory board meeting at the Hotel Coronado with 33 physicians, and I paid for all their tickets, all their hotels, and all their meals on my own Amex. They didn't pay me back my Amex bill. So I said, you can keep all of that money. I'll relieve you of the debt, but I want to take the bioelectric and the hydrogel and the pump IP that we started to develop with me. And I not only want to apply it to the heart, but I want to look if maybe the same idea would work for other organs. And that was the birth of Leonhardt's Launchpads and our Innovation Accelerator. So to shorten it up again, we try to fix everything with non-invasive bioelectric stimulation first. And only if that doesn't work, do we look at injecting biologics and stem cells. We just finished a 105-patient erectile dysfunction study. We applied our signals for stem cell homing, for arteriogenesis, for muscle regeneration and nerve regeneration non-invasively. 90% of the 105 patients were satisfied with their results 6 months after stimulation. And we only stimulated a few minutes twice a week for 4 weeks, a month out, 3 months out, 6 months out. They still had the function of that organ to a satisfactory level without having to use Viagra or anything like that. And so that typifies our approach. So that means we failed 10% of the time. For those 10% where we failed, we think those are candidates to go into the more sophisticated treatment that will be a combination of bioelectrics and biologics. So we're actually going to bring those patients back that we failed. We're going to give them another treatment, but this time we're going to add the injection of stem cells, exosomes, amniotic fluid, kind of a mix of regenerative proteins and stem cells, which has been studied in animals by us with Dr. Nestor Gonzalez-Cadiva at UCLA. At least part of that has. Sorry for that long explanation, but the other way to say it is every application we have in the accelerator is the same stimulator. It often is the same programs. Almost everyone has stem cell homing. Almost everyone has arteriogenesis. And we just call it something different when we point it to a different organ. Liver cells for liver regeneration, pancreas cells for pancreas regeneration. So it seems from the outside like we're trying to do too many things for a small organization, but really it's just one thing. And we just point it to different organs and we call it something different when we point it to that different organ.
No, it's look, I'm have hung out in a similar domain for several years now, and it's very elegant. I mean, as you were going through with some of these organs, these different biofactors and growth factors and so forth that need to be sort of in this sequence of events, I think you point to a very important thing that I think we've lost a little bit of, most definitely in sort of the pharma side of things, where we think everything's a silver bullet nowadays. But when it comes to these areas like complex regeneration, this isn't a single magic bullet. You got to really think through, as you guys have, what cells go where, what factors, what timing of these factors, and so forth. It's quite elegant. And, as I said, you have, quite a bit of projects here. You mentioned a bunch of them. If I could just ask you for a couple of minutes, can you talk a few minutes about the stem cell bra? Because this is an area that I just happen to know from previous careers. It is a multi-billion-dollar space, and I was just wondering, how's everything going along there? Because it's so lucrative.
Yeah, thanks. It always kind of gets some kind of reaction from people. Stem cell bra is designed to be a bioelectric bra that homes your own stem cells and increases circulation in the breast tissue. We've completed 16 large animals over a 3-year time period. We did histopathology on these animals. We were really scared that if we brought this in human clinical trials, we wouldn't want to do any harm. We wouldn't want to cause abnormal growths or cancer. So we were very careful, and the histopathology came out very good. No teratomas, no unusual growth. Those 16 animals had an increase of their breast volume about 20 to 30% with stimulating 1 hour a day, 1 hour every other day for 4 weeks. We then moved to a safety study in humans where we turned down to only a half hour once a week for 4 weeks. We didn't want to go for the gusto, and we just wanted to make sure everything was safe. We got through that safety trial, and we had no adverse events. We also didn't get any breast growth. So we're kind of thinking that a half hour once a week instead of 1 hour every other day was going to get some kind of increase of breast size. It didn't turn out, but we were glad that we took the safe approach and documented a safety study that was below the threshold of efficacy. Sure. And now we're getting ready with a few more animal studies done just to double check safety. And we've added this Clotho signal, so we had to go back and repeat some of the studies. And we're getting ready to do our first efficacy study. A center in Spain has signed up to do that study, and we'll report the results to everybody soon. Another part of stem cell bra is augmenting fat graft transplantation. Okay. We met with Dr. Joel. His last name slipped my mind right now, but he was the doctor that did Suzanne Summers, who was had breast cancer. She had a fat graft and stem cells to recover her lost breast after cancer, and he wants to use the bioelectric stimulation to augment that procedure.
Mm.
And a study just was published in Denmark that showed augmenting fat grafting with stem cell transplantation has less relapse, meaning a problem with these patients that get fat grafts is one breast will be about 20 to 30% smaller than the other because the fat kind of shrinks after they put it in. It's kind of a crude way of saying it, but that's basically what happens. And so everybody in the field is trying to strive to reduce relapse or reduce loss of tissue after the fat grafting. And Dr. Joel Aronovitz of Cedars-Sinai and UCLA, he wants to use this as a combination with fat grafting, but he only wants to do that after we finish a few more studies that we're doing that I mentioned earlier, and then we're going to apply to the FDA and the IRB to get started at Cedars-Sinai.
Excellent, excellent.
So we don't have that grand story that we have a bra that increased breast size 30% yet, but it may be a story that we can share with you a year or two from now. But we're proceeding very cautiously because we know
Certainly.
that this is a very could be a very serious safety issue if it was done wrong.
Certainly, certainly. I'd love to transition now to your long history as an investor in the space. You mentioned some of with your IPO with Bioelectric and so forth in the past, and I've been around the pharma industry about 30 years myself now. Have raised money sort of the old-fashioned way in the past, which was sort of this typical path of angel, your venture capitalists, your crossover investors, and then on to the public. It's a much different world nowadays in 2021. We have these gigantic angel rounds from venture capital groups, hundreds of millions of dollars. We have crypto over here. We have the crowdfunding. Give us your lay of the land in terms of sort of where we are in 2021 with bioventure funding in general, sort of some of the pitfalls, some of the areas that you think are exciting for young companies to look at. Walk us through a little bit on your experience here.
It's a big question, and I'm still learning as we go along. We primarily have used angel funding and family funds. We have had corporate investing with Bioelectric. Guidant invested 4 times. Boston Scientific invested. Abbott made a $900,000 license with us, which was like an investment. J&J agreed to underwrite 20% of the cost of our clinical trial if we made it mandatory to use their color mapping system in the study, which we did. So we have a combination of those 3 things. We don't seem to fit the VC model. Part of it is that the Leonhardt's Launchpad's business model is that Leonhardt Ventures maintains 50.1% control of each invention up to exit. We do waive that in a few occasions. We waived that with Second Heart and a few others. The 38 innovation assets or startups in the accelerator right now, 34, we have 50.1% control, and we've given up control on 4%, which we don't like. The only thing I found that really works for a starting entrepreneur is to build a very strong advisory board, to reach out to the opinion leaders in the field, bring in advisors that are opinion leaders, and basically ask each member that somehow engages your dream and your vision to try to find 3 people in their life network. It could be their brother, their cousin, their neighbor, their college friends, but usually every person that has had good relationships with people over the years has at least 3 people that will take a risk on something that they recommend. And we also make sure to tell everybody that what we're doing is very risky. It's maybe slightly better than going to Las Vegas and putting it on the roulette wheel, but we don't want anyone to take their nest egg or anyone to ever think that just because we're excited about the early results in a pilot study that this means some kind of guarantee for investment success. This is truly gambling, and it appeals to some people and it fits into some people's portfolio, and for others it's just absolutely the wrong thing to do because we're trying to do things that no one has ever done. No one has ever successfully regenerated a heart. No one has ever successfully regenerated a pancreas, and it doesn't mean that it shouldn't be done because it's hard to do, but it means that it's very likely that we may fail. The last 6 drugs with over $300 million spent for treating class 3 heart failure, particularly diuretic resistant patients that are retaining fluid and can't get rid of their excess fluids,
They have failed despite $300 billion being spent. So these things were, and then we come along with our modest $3 million budget, and we think we're going to do better than 5 companies that have failed with 300 billion. But yes, we do think that, but it is a little bit crazy to think that. And we're taking in the misfits and the other people that, you know, kind of have this crazy dream that this might be possible. Now the rewards can be great if it works out, but the only other thing I'd like to say that it would, you know, it's a long subject that could take many angles, but one of the reasons we have the Innovation Accelerator, a portfolio of innovations, if I've learned anything about investing in this sector over the years, is you have to have a portfolio.
Sure.
There's no one so good that you can pick one stock and be absolutely right on that one stock, or be absolutely right 100% of the time. When we started the Leonhardt launch pads in 2008, we were inside the University of Northern California Science, Technology, Innovation Center, UNC STIC. Well, I was a director of UNC STIC, and at the same time, Leonhardt launch pads, and we would bring in some technologies from the outside. And UNC STIC, led by the late Dr. Y. King Liu, we brought in Saffian, Ossian, and Tissuegen. And we evaluated a whole bunch, and we decided, you know, by the way, I joined that board in 2000, and every year I'd come to the board meeting and I said, we need an incubator. At that time, the word accelerator wasn't in vogue. And
Sure.
they would say, no, no, no. And then finally in 2007, Dr. Liu said, let's give it a try. So they gave me a $400,000 budget. So I used like $100,000 to buy some equipment to set up a laboratory, and then I invested $100,000 of their money in 3 different companies, Ossian, Tissuegen, and Saffian. And then we assigned PhD students from the school to work on the projects. Our professors helped. It was, and the only reason I got the board to accept it is they said, as long as you guarantee that one of our PhD students will be assigned to the project. So when people came into the incubator, I said, you have to accept that one of our PhD students is going to be assigned to your project. They'll sign a thing that says the IP belongs to you. I thought Tissuegen, which had electroacupuncture for knee pain, was going to be the success. The other company, Ossian, they took the catheter that we had developed for injecting stem cells in the heart, the MyoCath catheter, and they asked if they could use that for injecting bone cement in spines. And I said, sure. We had never thought of injecting bone cement with that catheter. We designed it for stem cells for the heart. So they took that and they developed a company around that. And then Saffian had the idea of injecting superglue in legs, primarily of ladies, to treat varicose veins, and basically to have the blue vein appearance go away from the skin and go deeper so that their legs don't have those ugly blue veins. And I thought that had no chance of success, but we took it in because Don, the founder, Don Crawford, I think his name was, I know his name was, he was just such a nice guy with such passion. But when we had the first meeting internally, I was, how are we going to build a business? There's no IP. You don't own superglue. It's a straight catheter, and they're using their thumb to smash it down. Why would any company buy that? Because it's eminent. It's the domain of everybody. You don't own such. So we tried to build some IP. We reduced the heat and the toxicity of the cyanoacrylate. We developed some fancy tip catheters, and we built an IP story. And that sold for $238 million to Medtronic in 2014. The $100,000 that I put in from the fund returned $6.3 million back to the University Foundation. Tissuegen, the one that I thought was going to be successful, still hasn't made a return. Dr. Liu decided to study it in his own clinic at the university. And when we went to go present the data, everybody said, this is your company, your invention, and your clinic did the study. We don't believe the data. Go do the study at an independent clinic. So that was a learning lesson there. Don't ever do a study of your own product in your own clinic, because no one wants to believe the results. Even though Dr. Liu was like, why go somewhere else? I have a clinic here. It was a bad idea. Ossian sold to Merit Medical, and I can't tell you what it was because it's an undisclosed amount. But I can tell you that the UNC Foundation ended up putting $1.1 million in. More than 7 years later, upon the sale to Merit Medical, we got a $1.1 million check back. And we were kind of astounded. And we learned from that lesson that when you sign with VC financing, there's liquidation preference, there's warrants, and somehow the guys who came in the last 3 years before the sale took the entire prize of the sale, and all the original investors and the founders and the people that did the work scrapped by with barely getting back what they had put into it. And that kind of made me think twice when I opened up the new class about whether we would take VC financing. And by the way, Safian was primarily angel investor financed. There were some VCs that came in, but they were on better terms. And there, 100,000 became 6.3 million. You know, so there's an example where I'm supposed to be an expert. I thought Tishigen was where I was going to put all my money. That ended up not providing any return at all. The one that I thought as an expert had no chance of success ended up being a success. So all I can say is, no matter how smart you think you are, you're better off having a portfolio, because it's so hard to be so good. It's like baseball. It's hard. The best of the best are lucky if they hit that ball 1 out of 4 times.
Oh yeah, isn't the truth? No, I appreciate you going into the uniqueness of this space in terms of the investor dynamics, and you know, as much as you focus on science, what you have to deal with in keeping these projects moving forward. And as you know, as you have a portfolio of 30 things, it's I take my hat off to you that you balance all this. Very impressive. Howard, any other, obviously nothing confidential, but any other big news coming up, talks, conferences you're going to be at, anything that?
I just wanted to say a few things about cancer.
Sure.
We were trying to stay absolutely focused on the convergence of bioelectrics and biologics for organ regeneration. But while we were trying to grow blood vessels, we found some signals that actually cause blood vessels to retreat away from the organ. And that was looked to be a failure for organ regeneration, but we thought, oh, maybe it would be nice to get blood vessels to retreat away from tumors. So that started us filing our first bioelectric cancer patents. And then we were basically trying to proliferate stem cells, and we found some signals that actually caused apoptosis that stopped them from dividing. Dr. Genovese called me one day and I said, well, how did those signals work? And he said, oh, terrible. Complete failure. The cells were multiplying. We put those signals in the dish and the cells stopped multiplying immediately. And he said, we failed. And I said, well, listen to that. There might be some applications where you would want cells to stop proliferating. Let's maybe file this signal for cancer treatment, and let's start doing some studies. So we've done some animal studies at UCLA that did not work. We did not cure cancer in mice in our first try. But now we have 9 issued patents for cancer treatment, for bioelectric cancer treatment, including customized reading of a tumor, and then bioelectrically signaling the tumor to change its surface proteins to elicit immune response and its surface charge, to starve it of blood supply, and to also basically reprogram it to stop dividing or cause apoptosis. And we're about to go into another round of animal studies, but we really do believe that despite the fact that our first mouse study didn't succeed, that we are on to something. And while we were doing this, 2 other companies, Novacure and Pulse Biosciences, and a third one called Automedical, they've focused just on this, and they've gone a little bit further than us. And Novacure, from a business point of view, was only worth $24 million 5 years ago. And the last I look, I think they're trading at $14 billion. And they're using bioelectric treatment or electromagnetic treatment to treat glioblastoma, one of the most aggressive forms of brain cancer. And they've had success, not perfect success, but better success than chemo and radiation. And all of us, and my mom passed away of cancer, and everyone that's listening knows somebody. And that chemo and that radiation really ravishes these people when they're already weak and sick. And if we can possibly make a contribution to be just as effective without those side effects of chemo and radiation, that alone will be a big contribution. So I'm really excited about the possibility that in the future, and with Novacure and Pulse Biosciences both having clinical successes and commercial successes. When we first brought the idea of bioelectrically treating cancer, just like when we first brought the stent graft idea, everybody thought it was crazy. Everybody thought, if it's so good, why isn't everybody else doing it? everybody thought it was, out in left field or in outer space. And now that it's been proven in 3 different studies, we're looking less crazy. And I really believe that it's going to be a big part of our legacy as an organization, that we are going to make a contribution to cancer treatment.
Yeah, most definitely. And I just a side note I usually bring up on Regeneration shows, it's a bit of a paradox, but we don't normally think about the fact that the most cancer resistant organisms on this planet are the regenerators. And so there are some very interesting secrets, whether it's amphibians or jellyfish or starfish. There's a reason why they are so good at taking cancer and turning it around into normal tissue. So I think you're definitely on the right path there as well. Howard, it's really it's fascinating listening to you, and as I was saying, 30 plus projects in the portfolio. I take my hat off to you. I wish you the best with all of this. Obviously in biotech, some win, some don't, but you clearly, based on your portfolio concepts, you're on the right path here, and it's just, really rooting for you. For everybody that's going to be listening to this show on our podcast or watching on the YouTube channel, you've been listening to Howard Leonhardt, founder of Leonhardt Ventures, working at the convergence and intersection of bioelectrics and biologics for complex organ generation, healing, cancer reversion, ultimately. Howard, it's always great seeing you. I want to thank you for taking the time out of your schedule to talk to us for a little while. Thank you for everything you have going on there. And as we say on our show, thanks for helping to create a better tomorrow through all these technologies. It's a really very fascinating topic.
Thanks, Ira. And you did touch on the social good impact portfolio. I just wanted to make one mention of that.
Sure. Take it away.
Basically, the family foundation, Leonhardt Family Foundation, that makes some money from this biotech stuff. We made a commitment to reinvest a substantial part of that into social good impact. And I got to be part of the Conscious Capitalism Society and have dinner with John Mackey, one of the co-founders, the founder of Whole Foods. And he basically pulled me into this idea that, when we had some successes in the past, we did our philanthropy. We gave almost $1 million to the University of Florida. We gave money to Florida International University. We gave money to the Ohio Cell Therapy Foundation. But he convinced me that, and the society really has this charter of, it's better to help people to learn to fish than just to give them a fish. And the idea that invest in social good impact companies that can be sustainable, can keep doing good. So that's always going to exist. We did have that mixed into the Cal-X Stars Business Accelerator, but after a while, the board said it was too confusing. So we separated the portfolio of social good impact to Cal Impact, Social Good Impact Accelerator, and Leonhardt's Launchpad is focused on just the convergence of bioelectrics and biologics. And that way investors don't get confused about the two things being mixed. And the simple story is we're going to take we're hoping to sell some of our assets. We've actually begun selling some of our biotech assets, and we're going to plow some of that back into the social good, which includes a platform, as you mentioned, for helping people that are having difficulty in raising capital to have a more efficient pathway to do so.
Outstanding. Really, really great story, Howard. Thanks so much for telling about it.
Thanks, Ira. Great to see you again.
Good seeing you.